In this chapter, we're going to be learning about metabolism.
Metabolism is the different chemical reactions that are going to happen in our body to allow us to obtain and use the energy from the food we eat. So if you remember, we said that nutrients like our proteins, carbohydrates, and lipids contain calories, and what that means is they contain energy. Calories are a measure of energy. Now, what metabolism is telling us the different steps that our body needs to go through to get the energy from our protein, carbs, and lipids and use the energy for our body's needs. So that's basically going to be what we're going to be looking at in this chapter. Now, before we get started, there are a few new terms that we want to review, starting off with the terms anabolic and catabolic. These are just describing two different types of reactions. Anabolic reactions are ones where we are building different structures. So, for example, if we look at our proteins, it's showing us that when we put together the individual amino acids, we make a larger protein structure. And since we are building, this is referred to as an anabolic reaction, and anabolic reactions actually require energy. So if you see over here, there's an arrow going inwards that says uses energy. That's because in order for us to build, we need energy. So anabolic reactions are where we are building something, and this requires energy. On the opposite end, we have our catabolic reactions, and these are reactions where we are breaking something down. So, for example, we're taking our protein structure and breaking it into smaller pieces or individual amino acids. So, since we are breaking it down, it is called a catabolic reaction, and when we perform catabolic reactions, it actually releases energy. So, again, just to recap, anabolic reactions are building and they require energy. Catabolic reactions are breaking down and they release energy. Now, this energy that we've been referring to is going to be stored in a structure called adenosine triphosphate, or ATP. ATP is usually referred to as our body's energy currency, and basically what that means is that ATP is the way that we measure or count how much energy we have in our body. So, for example, in the US, the way we count how much money we have is by counting in dollars. With our body's energy, the way that we count how much energy we have is by counting how much ATP we have. So that's kind of what it means when you hear that ATP is our body's energy currency. That's how we count and measure the amount of energy in our body. Now, the reason for this is because this is the structure where energy is actually stored. So, we said it is called adenosine triphosphate. That's because it's made out of adenosine and then 3 phosphate groups. That's why it's triphosphate. And if you take a look, the phosphate groups are attached using this bond right here. And this bond actually has energy stored in it. That's why they have it in red, to signify that there is energy there. So the energy is actually being stored in these phosphate bonds. And so if we wanted to use energy, what we would do is break off this phosphate group, and then the energy that was in its bond will go ahead and be released, and we can use it in our body for whatever we needed it for. If we needed more energy, we can go ahead and break off this next phosphate group, and that will release the energy that was in its bond, and we can go ahead and use that energy as well. Now, that being said, after we use up the energy, after we've broken off these phosphates, we can go ahead and regenerate the ATP structure and be able to store energy there again and use it a second time around. And we're going to go ahead and take a look at how that happens. Now, this here shows us how this process is done when we are using up our energy and then regenerating it. So here you see we have our ATP structure, the adenosine with 3 phosphate groups, and we needed to use some energy, so we went ahead and broke off one of those phosphate groups. That released the energy for us so that we could use it, and as you can see, now that we've removed that phosphate group, we have ADP, adenosine with 2 phosphate groups added to it. Now, The next thing that we're looking at here is, what if we made more energy and we wanted to go ahead and store it? All we would have to do is add that phosphate group back on, and the energy that we have that we want to store will go ahead and store in the bond that attaches our phosphate for us. So again, we used up some energy and broke off the phosphate group, but if we have more energy that we want to store, we'll just add a phosphate group back on and store the energy in its phosphate bond. And now we've gone back to having adenosine triphosphate, the three phosphate groups, and we can go ahead and use it again. So again, this is just briefly showing us how once we use our ATP, we can regenerate it and store energy in there again. Now that we've covered the new terms that we wanted to review, we can go ahead and get started with our actual metabolism pathway. Now, we're going to have two different pathways that we are going to look at. We have our anaerobic and our aerobic pathways. And before we get into each one of them, I just want to talk about the differences between them. Now, the primary difference, the obvious difference between them, is the aerobic metabolism is going to require oxygen. So aerobic means requiring oxygen. Anaerobic means it does not require oxygen. So the first difference, aerobic is the pathway that requires oxygen. Anaerobic does not require oxygen. Now, whenever a reaction requires oxygen, it needs to move into the mitochondria. So that means that all of our aerobic pathway steps will be in the mitochondria. If it doesn't require oxygen, it can happen in the cytoplasm. So the anaerobic pathway will be in our cytoplasm. The other difference between them is that the aerobic metabolism actually makes so much more ATP. Anaerobic only makes about 2. Aerobic makes over 30 ATP. And for that reason, it's usually our body's plan A. The body prefers to do aerobic. If it can't do aerobic, then it will do anaerobic, or in certain situations, the anaerobic might be beneficial. For example, one of the other differences between aerobic and anaerobic is that the aerobic pathway is a lot longer. It takes a lot more time than the anaerobic pathway. It's able to make so much more energy, but it does take its time in making that energy. Now, if you were doing something that required energy quickly, you needed that energy quick. For example, if you were doing really high intensity exercise, you're using up that energy really quickly per second. And so in that kind of situation, you need energy to be replaced quickly. You don't have time to go through the long steps of the aerobic metabolism, and so in that situation, you would prefer the anaerobic pathway, because even though it doesn't make as much ATP, it gives you that energy quickly. So again, anaerobic doesn't make as much ATP, but it's a very short, quick cycle, so it's going to be beneficial in situations where you are using up your energy very quickly and you need it replenished very quickly, like if you were doing high intensity exercise and using up lots of energy per second. Now, if, for example, you were just taking a stroll in the park, you are not using up much energy per second, you are using your energy up a lot more slowly, so you don't need it replaced as quickly. In that situation, you're going to go with plan A because it makes so much more energy, the body prefers it, and we have time to go through this long pathway and end up producing all of that energy in the end because we're not using our energy up very quickly when we're doing something like, for example, just taking a stroll. That doesn't require much energy, so we don't need to replace it very quickly. It's not going to run out quick. We have time to do our aerobic and end up with lots of energy in the end. So again, just to recap the differences between them, aerobic requires oxygen, so it's going to happen in the mitochondria. Anaerobic does not require oxygen, so it will happen in our cytoplasm. The aerobic is our plan A because it makes so much more ATP. And because of this, it also takes a long time. So it makes a lot of ATP, but it's a long pathway to end up with all this ATP in the end. Anaerobic only makes about 2 ATP, but it is a shorter, quicker cycle. So in certain situations where we need energy quickly, we will go ahead and go with our anaerobic pathway. Now, since the anaerobic pathway is a lot shorter, we're going to start off with that one first and get it out of the way, and then we'll spend the rest of the time on the aerobic pathway. Now, glycolysis is always going to be our first step. No matter which pathway we take, we're always going to do glycolysis first. And after we perform glycolysis, that's when we decide if we are going to take the anaerobic or aerobic pathway. So again, before we decide which pathway, we're going to do glycolysis first. Glycolysis does not require oxygen, so it'll actually start off in the cytoplasm. So our metabolism will always start in the cytoplasm with glycolysis. After glycolysis, we then decide, are we going to stay in the cytoplasm and choose the anaerobic pathway, or are we going to move into the mitochondria and start the aerobic pathway? So again, glycolysis is always the first step, and then we decide which pathway we're going to take. Technically, all glycolysis is the conversion of glucose into pyruvate, but let's go ahead and take a closer look at that process. So this over here is showing us the process of glycolysis. I know there's a lot on here. I don't need you guys to know all of it. I'm going to go ahead and point out to you the few things on here that I do want you to pick up. So glycolysis, we said, is the conversion of glucose into pyruvate. So glucose is a structure that is made of 6 carbons. So that's what we're starting off with, a 6-carbon structure, which is our glucose. And what's going to happen is we are actually going to split this glucose structure in half. So that's what you see happening over here. So again, I want you to start off with step 1. We begin with glucose, and then jump over to step 2, where we are splitting that glucose into 2 units. So now what we have is 2 3-carbon compounds. Instead of 1 6-carbon compound, it's split into 2 individual compounds, each having 3 carbons each. Now, these 2 compounds are going to go through a few different reactions. I don't need you guys to know exactly what's happening in those reactions. I just want you to know that after we split it, we take these 2 carbon compounds and put them through a few reactions, and in the end, they end up as 2 units of pyruvate. Pyruvate is another structure that is made out of 3 carbons. It has other things added onto the 3 carbons that make it different than what we started off with in step 2, but I don't need you guys to know all of those details, so I'm just going to streamline it for exactly what I want you to know from here. So with glycolysis, we start off with a 6-carbon glucose. It's split in half into 2 3-carbon compounds. Those 2 3-carbon compounds go through a few different reactions and then become 2 units of pyruvate. Now, in this process of glycolysis, we end up making 2 units of ATP. So, glycolysis is starting off with 1 glucose unit and ending up with 2 units of pyruvate. And what we also make in this process of glycolysis is 2 units of ATP. Now, another thing that I want you guys to notice is over here where it shows you coenzymes and then hydrogens and electrons. This is going to be a picture or a part of the diagrams that you're going to see over and over again as we go through all of the steps. And this is because every time we break something apart, we are releasing hydrogens and electrons. And we have these coenzymes that are going around and collecting these hydrogens and electrons. And the reason they're doing that is because they can actually cash them out for energy at the end of our cycle. So again, these coenzymes are kind of like these little shopping carts going around collecting all of the hydrogens and electrons so that they can take them to our cashier at the end, which is the electron transport chain, and cash them out for a bunch of energy. So we can actually cash out the hydrogens and electrons for energy in the electron transport chain. Now, that being said, this cashier that's going to cash us out for energy, the electron transport chain, this is only present in the aerobic pathway. It's not present in the anaerobic pathway. So right now, we don't know which pathway we're going to take yet. Remember, we said we do glycolysis first, and then we decide which pathway. So since we're still not sure, we're going to go ahead and collect these hydrogens and electrons anyway, just in case we take the aerobic pathway and can make our way over to this cashier, the electron transport chain, and cash them out for energy. If we end up taking the anaerobic pathway, then the coenzymes can't really do anything with the hydrogens and electrons. There is no cashier at the end of the anaerobic pathway, and so we wouldn't be able to cash them out for energy if we chose anaerobic. But right now, we haven't chosen which pathway we're going to take yet, so those coenzymes are going to collect those hydrogens and electrons anyway, just in case we decide to take the aerobic pathway. Now, at this point, pyruvate is going to need to decide, is it going to stay in the cytoplasm and go the anaerobic pathway, or is it going to move into the mitochondria and take the aerobic pathway? Depending on which pathway it takes, it's going to convert into a different substance and take different steps from there. We're going to go ahead and start off with our anaerobic and take a look at what happens to pyruvate in that situation. Now, when we choose the anaerobic pathway, remember we said that we were collecting those hydrogens and electrons just in case we chose the aerobic pathway and could cash them out for energy in the end. But since we chose the anaerobic, we can't cash out those hydrogens and electrons for energy. We don't really have anything that we can do with them. So basically what happens here is the coenzymes are going to give the hydrogens and electrons back to pyruvate because they realize we can't do anything with them. Since we chose anaerobic, there is no cashier at the end of this cycle that can cache these hydrogens and electrons for energy, so we don't need them anymore. So we're just going to give them back to pyruvate. So that's what's going to happen next. If we chose anaerobic, the coenzymes are just going to give the hydrogens and electrons back to pyruvate because they realize they can't do anything with them. And that's what you see happening over here. This structure that you see over here where it says NADH, NAD, this is actually the name of the coenzyme that we were looking at in the beginning, the coenzymes that we said were collecting our hydrogens and electrons. Technically, the name of the coenzyme is NAD. When it collects those hydrogens, it is NADH. So what you're seeing over here is this was the coenzyme carrying our hydrogens and electrons. And what it actually did is it gave the hydrogens and electrons back to pyruvate. And so now it's only the coenzyme by itself now. It doesn't have the H on it anymore because it gave the hydrogens back. So again, NAD is the coenzyme that was collecting hydrogens and electrons. Since we chose anaerobic, it knows that we can't cash those out for energy, so it's just going to give them back to pyruvate. And this over here was our pyruvate structure. Remember, we said it's a 3-carbon structure with other things attached to it. So this is pyruvate, and if you take a look at what we have over here, this structure is nearly identical to pyruvate, except for the fact that it has these added hydrogens onto it. So lactate is actually what we end up with after the coenzymes give the hydrogens and electrons back to pyruvate. So one more time, when we are choosing our anaerobic pathway, the coenzymes can't do anything with the hydrogens and electrons, so they are going to give pyruvate the hydrogens and electrons back. When pyruvate accepts the hydrogens and electrons from the coenzymes, it actually becomes a structure called lactate. So lactate is just pyruvate with the added hydrogens and electrons onto it. Now this over here is showing us the larger picture of this process. So this is our anaerobic pathway. Another name for the anaerobic pathway is the Cori cycle. And we've seen this part happen already. We went through our step of glycolysis, which was glucose to pyruvate. We decided to go the anaerobic pathway, which means we can't cache those hydrogens and electrons for energy. So the coenzymes gave them back to pyruvate. And when pyruvate accepted those hydrogens and electrons, the new structure is now called lactate. Now, at this point, we can't do anything else. With the anaerobic pathway, it basically stops there. We don't have anything else that we can do. Remember, we said that the anaerobic pathway only makes 2 ATP. But remember, that glycolysis step from glucose to pyruvate made 2 ATP already. So that's where those 2 ATP come from. So again, the anaerobic pathway only makes 2 ATP, and where it gets those 2 ATP from was from the glycolysis step, the glucose to pyruvate. That made 2 ATP, and that's all the energy that the anaerobic pathway can make. So since the anaerobic pathway can't do any more at this point, one thing that it can do is try to get us to regenerate glucose so that we can perform glycolysis again. Because again, every time we perform glycolysis, we make 2 ATP. So the purpose of the anaerobic pathway, or what its goal is to try to regenerate glucose and force our body to do glycolysis again so that we can make 2 ATP. And then we're going to come around again, regenerate glucose, go through glycolysis again, make another 2 ATP. So again, we're just concerned about regenerating glucose over and over and over again, because every time we're back at glucose, we can perform glycolysis again and make another 2 ATP. And that's what we're seeing here. So we ended up with lactate. Again, we can't go any further now since we're anaerobic. That's as far as we can go. So what we are doing instead is wanting to regenerate glucose so that we can perform glycolysis again and make another 2 ATP. Now, the issue is that we can't reverse this process and regenerate glucose except for in the liver. So if we want to regenerate glucose, we need to send our lactate to the liver, and then the liver can reverse that process where we reverse from lactate back to pyruvate, and from pyruvate back into glucose. Now that we have generated glucose again, we can perform glycolysis again and make another 2 ATP. So again, just to recap, the anaerobic pathway, or the Cori cycle, is only going to make 2 ATP. We make those 2 ATP in the glycolysis step. After that, pyruvate will accept the hydrogens and electrons from the coenzymes, and that will create lactate. We can't go any further at this point, so what we want to do is reverse the process and regenerate glucose so that we can go through glycolysis again and make another 2 ATP through our glycolysis step. In order to regenerate glucose, we need to send lactate to the liver. In the liver, lactate can be converted back into glucose so that we can start the glycolysis process again and make another 2 ATP. Now, this here is showing us the same thing, just in a little more detail. So you see we started with glucose, we made pyruvate. When the coenzymes gave the hydrogens and electrons to pyruvate, what we ended up with was our lactate structure. And then it's showing you that we send lactate to the liver, and the liver can convert lactate back into glucose so that we can go through this process over again. All right, so that covers our anaerobic pathway. Again, it's just glucose to pyruvate to lactate and reversing over and over again. So now we can go ahead and take a look at the aerobic pathway. Now, if we are choosing the aerobic pathway, pyruvate is going to need to move into the mitochondria first, because remember, the aerobic steps can only occur in the mitochondria. So since we chose to do aerobic, we are going to move pyruvate into the mitochondria. And then at this point, we can go on with our next steps, and I'm going to go ahead and walk you through that using our diagrams. So here it's showing us what would happen to our pyruvate structure. So remember that the only reason that we gave back the hydrogens and electrons was because we chose anaerobic, and with anaerobic we do not have our cashier to cash them out for energy. Now we've chosen aerobic, which means we can have those hydrogens and electrons cashed out for energy. So because of that, we're going to continue collecting our hydrogens and electrons. We're not going to give them back to pyruvate. Remember, pyruvate only became lactate when it was given the hydrogens and electrons back. It's not going to take those hydrogens and electrons now, because the coenzymes want to keep collecting them because they can actually do something with them. So because of that, in the aerobic pathway, pyruvate is actually not going to be converted into lactate. It's not going to be getting those hydrogens and electrons from the coenzymes. The coenzymes are going to hold on to them and keep collecting them. Instead, what's going to happen is, again, pyruvate is made out of 3 carbons. In the aerobic pathway, one of the carbons is going to be removed off of pyruvate, and the remaining 2 carbons are going to attach to a coenzyme called coenzyme A. And the structure that we end up with is the 2 carbons from pyruvate with coenzyme A attached. This structure over here is what we call acetyl-CoA. So again, in the aerobic pathway, again, we're still starting off with pyruvate, but instead of it being converted into lactate, it's going to be converted into acetyl-CoA this time. The way we do that is we remove 1 carbon off of pyruvate, the remaining 2 carbons attach to coenzyme A, and that creates our acetyl-CoA structure. Acetyl-CoA can then go ahead and move on to the next step of our aerobic pathway, which is the TCA cycle. Now, this over here is showing you what we just covered, but it is reminding you that glucose actually makes 2 pyruvate units. So the picture that we just saw was just showing us what happens to 1 pyruvate, but this is just a reminder that we're actually going to be doing that process twice, because with glucose we actually make 2 pyruvate units, so both of those pyruvates will end up being converted into acetyl-CoA. Alright, now before we move on to our TCA cycle, I know I said that our acetyl-CoA will enter into the TCA cycle, and we will look at the TCA cycle, but before I get into that, I want to show you guys where our other nutrients come into play, because so far all that we have seen is carbohydrates. Because we started off with glycolysis, glucose to pyruvate, and we know that glucose comes from our carbohydrates. So the pathways that we've looked at so far has only really been for the carbohydrates. We haven't seen how our proteins or fats would enter metabolism, and they also produce energy, so we do need to see how they would enter metabolism and be used to make energy. So let's start off with looking at our proteins. Proteins are made of amino acids, and amino acids can either be glucogenic or ketogenic. What glucogenic means, glucose stands for glucose, genic stands for the creation of. So literally what it means is the creation of glucose. So amino acids that are glucogenic can be used to generate glucose. And if we follow the arrows, you'll be able to see how. So we have some amino acids that can enter metabolism by being converted into our pyruvate structure. Now, this step from glucose to pyruvate is actually reversible. And because it's reversible, this is going to be very important because it's going to allow some of our nutrients to be able to convert back into glucose. And that's exactly what's happening here. Since these amino acids are going to be converted into pyruvate, and pyruvate can reverse back into glucose, that means these amino acids can technically be used to make glucose. This is going to be beneficial in the situations where maybe your body doesn't have enough glucose and it needs it to use for fuel, and so it's going to have to make its own glucose. One of the ways that we can achieve this is through this pathway over here, using these glucogenic amino acids. Now, some of our amino acids cannot be used to make glucose, and these are going to be called ketogenic, meaning that they will make something called ketones instead, and we'll learn a little bit more about exactly what that means and how that's done later on in this chapter. But for now, let's go ahead and take a look at where these amino acids enter and why is it that they're not able to make glucose. So these set of amino acids enter into the metabolism pathway by being converted into the acetyl-CoA structure. If you take a look, acetyl-CoA is not reversible back into pyruvate. And the way that we would get to glucose is by going to pyruvate and reversing back up into glucose. Since the step from acetyl-CoA to pyruvate is not reversible, that is not possible. With the first set of amino acids, we entered where these steps were still reversible and we could go up to glucose. But acetyl-CoA is not reversible. So what that tells us is once we've reached acetyl-CoA, we can't reverse back up into glucose, which means that these amino acids are not glucogenic. They are not able to make glucose. Now we also have a third entryway that's not shown on here for you because it actually happens later in the pathway. So we're going to go ahead and move on to another picture that shows us that third entryway of our amino acids. Now this here shows us all three entryways. Again, these are still amino acids. They've just drawn them out for you now. So this was our first set of glucogenic amino acids that we said entered the pathway by converting into pyruvate. This is our second set of amino acids that we said entered by converting into acetyl-CoA. And the third set of amino acids that was not shown on the previous picture were these down here. And these amino acids are going to enter straight into the TCA cycle. Now, these that enter into the TCA cycle are actually glucogenic as well. And we'll learn a little bit why that is later on in this chapter. But for now, what I want you guys to know are the three different entryways for amino acids and which ones are glucogenic, which ones are not, and what that means. So the first set enters into pyruvate, and that is glucogenic, meaning it can be used to make glucose. The second set of amino acids will enter by being converted into acetyl-CoA. That cannot reverse back up, so it cannot be used to make glucose. The third set of amino acids is glucogenic, and it is going to enter directly into the TCA cycle. Alright, so that covers the protein part of our metabolism pathway. Now we can go ahead and look at how our fats are going to enter into the pathway. Now remember, we're talking about how we get energy from the nutrients in our diet. And if you remember from our fats chapter, we said that the fat that is typically found in our diet is in triglyceride form. Triglyceride is our main dietary fat, or the type of fat that is usually found in our foods. So that's what we're going to be looking at. And if you remember, triglycerides are made of a glycerol backbone and 3 fatty acids. So when we look at how our fats enter into metabolism, we're going to be looking at those two components, the glycerol and the fatty acids, because those are what are found in our triglycerides, and triglycerides are the type of fat that we have in our diet. So let's go ahead and take a look at how this would happen. So we said that we were going to look at glycerol and fatty acids because those are the components of our triglyceride or the type of fat in our diet. So let's start off looking at glycerol. Glycerol enters metabolism right before pyruvate. So remember, we broke glucose into 2 3-carbon compounds and then they were converted into pyruvate. So that step before they convert into pyruvate, that's where glycerol enters. Now, if you pay attention to this, glycerol is entering where the step is still reversible. So technically what that means is glycerol can be used to make glucose as well. But if we look at our fatty acids enter the metabolism pathway by being converted into acetyl-CoA. Now, that is something that is not going to be able to reverse. Remember, we said once we reach acetyl-CoA, this step is not reversible, and that means once we are at acetyl-CoA, we cannot be used to make glucose. So technically, these fatty acids, since they are being converted into acetyl-CoA, they cannot be used to make glucose. Now, another thing that I want you guys to pay attention to is the fact that this step right here is reversible. And what that means is fatty acids will be able to enter by being turned into acetyl-CoA, but acetyl-CoA can also be used to make fatty acids. This is important because we've discussed before that anytime we eat in excess, our excess is going to be turned into fat, whether it started off as carbohydrates or protein. When we eat too much of those, they'll be turned into fat. And this here shows us why that is. Remember that we were able to make acetyl-CoA out of our carbohydrates, we were able to make acetyl-CoA out of our protein, and now we've seen that acetyl-CoA can be turned into fat. So again, what that tells us is anything that can be made into acetyl-CoA can then be turned into fat. Since our carbohydrates and protein can make acetyl-CoA, technically they can be turned into fat as well. But let's go ahead and take a look at how these fats are entering in a little bit more detail. So over here, we are looking at the structure just drawn out a little bit more for us. So this was our glycerol, and it entered in between glucose and pyruvate. And this is an example of one of the fatty acids that could have been present. Now, if you remember from our fats chapter, fatty acids have a bunch of carbons attached to them. And if you remember what the acetyl-CoA structure looks like, we have it right here. Acetyl-CoA is made out of 2 carbons attached to coenzyme A. Since fatty acids have a bunch of carbons in them, that means that they can make a lot of acetyl-CoA. All we have to do is break off 2 carbons at a time, attach coenzyme A, and now we have made 1 acetyl-CoA. Then we'll take the other 2 carbons, attach coenzyme A, we've made another acetyl-CoA. And we're just going to keep doing that until we've used up all of the carbons in our fatty acid chain. Now, this fatty acid had 16 carbons, and as you can see, we ended up making 8 acetyl-CoA from that. Because remember, each acetyl-CoA is going to use 2 carbons. If we break off 2 carbons at a time, and we had 16, we end up with 8 acetyl-CoA. So we can actually make a bunch of energy just from 1 fatty acid for this reason. Now, another thing that I want to point out is we are still releasing hydrogens and electrons. This has been happening in all of our steps. Anytime we broke something off, there were still hydrogens and electrons being released. You might have noticed that on the previous slides where I didn't mention it, but you saw that it said coenzymes right there. This is just reminding us that anytime we break something off, we're releasing hydrogens and electrons, and the coenzymes are picking them up. So same thing goes here. Every time we break off these 2 carbons, we release hydrogens and electrons, and the coenzymes are there collecting them. When we break off our next 2 carbons, same thing happens. So just keep that in mind so that we can, again, build on that in a minute when we reach our electron transport chain, or the cashier where they will be cashed out. Now this here is showing us the process in just a little more detail. So we are looking at a fatty acid chain, and we know that acetyl-CoA is made out of 2 carbons with coenzyme A attached. So we are breaking off 2 carbons, adding coenzyme A. Now we have 1 acetyl-CoA, and the fatty acid chain is shorter. We can break off another 2 carbons, attach another coenzyme A. Now we've made our second acetyl-CoA, and the chain is even shorter. And we're just going to keep doing that until we have completely broken off or used up all of the carbons in our fatty acid. Now going back to this diagram over here, this is just a recap to remind us of what we've covered so far. And what I want you guys to notice from this is that the meeting place for all of our nutrients was acetyl-CoA. So whether we started off with carbohydrates or protein or fat, they all ended up making acetyl-CoA. So at this point we have a bunch of acetyl-CoA that we've made. Some of it came from protein, some from carbs, some from fat, but it's all in the form of acetyl-CoA now. And so we can go ahead and take this acetyl-CoA into our next step, which is the TCA cycle. So our next step was the TCA cycle. This is where all of the acetyl-CoA is going to enter next. Now, a few things that I want to mention about this. TCA cycle stands for the citric acid cycle, but technically it's also sometimes referred to as the Krebs cycle. It's the same thing. And the acetyl-CoA is going to need to enter into the cycle to continue metabolism, but before it can enter the cycle, it needs to bind onto oxaloacetate. If oxaloacetate is not present, acetyl-CoA cannot enter into the TCA cycle. Now, oxaloacetate is made from pyruvate, so that's where we are going to get our oxaloacetate from. But let's go ahead and take a look at the diagram to see exactly how this is going to happen. So this over here is showing us the TCA cycle. I know there's a lot going on here. I don't need you guys to memorize all of it. So I'm just going to point out to you what it is that I want you to know. So the first thing that I want to point out is oxaloacetate over here. And remember, we said it is made from pyruvate, so that's what you see happening. But you also see that oxaloacetate has a reversible arrow back into pyruvate. What that means is that oxaloacetate can also reverse back up into pyruvate as well. Now, the reason this is important is because it explains to us why the last set of amino acids was glucogenic. Remember, we said the third pathway for the amino acids was entering straight into the TCA cycle, and we said those were also glucogenic. Technically, when we say they're entering into the TCA cycle, the way they're doing that is by being made into oxaloacetate. So the third pathway of amino acids that had an arrow going into the TCA cycle was actually being converted into oxaloacetate. So now the amino acids have been converted into oxaloacetate, and since this step is reversible, the oxaloacetate can then reverse back up into pyruvate. But we also know pyruvate is reversible into glucose, so that's going to be how those amino acids can make glucose. So again, the last set of amino acids entered straight into the TCA cycle, and we said they were glucogenic. The reason they are glucogenic is because the way they enter the TCA cycle is by being converted into oxaloacetate. Oxaloacetate can reverse into pyruvate, and then pyruvate can reverse into glucose. So that's how we can make glucose out of that last set of amino acids. Now, that is just a side note, since I had mentioned that I would explain how that last set of amino acids was glucogenic. But let's go ahead and get back to the acetyl-CoA and how it's going to enter the cycle at this point. So we said the acetyl-CoA cannot enter the TCA cycle without combining with oxaloacetate. So here we have acetyl-CoA combining with oxaloacetate, and then they're going to go through the cycle together. Now, there are only a few things that I want you guys to pick up from this cycle. The first thing is to notice what is happening to the structure. As you can see, the structure is getting smaller and smaller, and that is because we are removing one carbon at a time as we go through the pathway. So the first thing that I want you to know is that when acetyl-CoA combines with oxaloacetate and enters the cycle, one carbon is going to be released at a time. And remember, anytime we break something off, we are also releasing hydrogens and electrons. And so that's why you see that over here. Hydrogens and electrons are still being released, and our coenzymes are still collecting them so that we can cash them out for energy. Now, when we release carbons, another thing is going to be released as well, which is our energy. Remember, when we break something off, that means it's a catabolic reaction, and catabolic reactions release energy. So, just to recap, acetyl-CoA is going to combine with oxaloacetate so that it can enter the cycle. As they go through the cycle, one carbon will be removed at a time. Since the carbon is being removed and released, what's also being released in the process is hydrogens and electrons. And since we are breaking something off and it's catabolic, we are also releasing energy as well. So I want you guys to know that oxaloacetate needs to combine with acetyl-CoA before it can enter. And I want you to know what is being released out of the TCA cycle. And that is carbons, hydrogens, electrons, and our energy. So that is the gist of what I want you guys to know from our TCA cycle. Now we have finally come to our last step that we have all been waiting for. We were collecting hydrogens and electrons this whole time so that we could reach our cashier at the end and cash them out for a bunch of energy. And remember, we said that cashier was the electron transport chain. So now we are at this cashier and we can go ahead and cash out those hydrogens and electrons for energy. So let's go ahead and take a look at how that would happen. So the electron transport chain actually occurs in the inner membrane of the mitochondria. And what's happening is those coenzymes that were collecting all of the hydrogens and electrons are going to come dump them into the electron transport chain, in particular into the inner compartment over here. Now, what we want is we want the hydrogens that were dumped into the inner compartment to actually move to the outer compartment, because if they're over here, they can come down using this pump. And when they go through this pump, that actually generates ATP. So again, the coenzymes will deliver hydrogens and electrons to the electron transport chain. They will be dumped into the inner compartment, but we want the hydrogens to move to the outer compartment so that they can come down using this pump and generate ATP. But the problem is that this outer compartment is more highly concentrated than the inner compartment, which means that it's going to take some energy to move the hydrogens up here. So that's where our electrons are going to help us out. The electrons are going to be able to generate some energy for us when they move down the membrane using these electron carriers. So we have these electron carriers in the membrane. As the electrons move through the electron carriers, that generates just enough energy to move our hydrogens across the membrane so that they can come back down using this pump and generate ATP. So again, let's recap one more time. The coenzymes are going to deliver hydrogens and electrons to the electron transport chain. They are going to end up in the inner compartment, but we want them to come to the outer compartment so that they can use the pump to make ATP. That is going to require energy, so the electrons will move down the membrane using electron carriers, because that will generate enough energy to move the hydrogens to the outer compartment where they can come back down using the pump, and that will generate ATP. Now, this pump over here has a few different names. You might have heard of it before as ATP synthase because it makes ATP, but for the purpose of this class, we're just going to refer to it as the protein complex pump. Now at this point we have completed all of the steps of metabolism. This here is a good diagram to look at. It shows you all of these steps together, starting off from the very beginning all the way down to the electron transport chain.
Everything we've learned so far about metabolism has been about in a regular state of eating, but now we're going to take a look at how things change when you overeat, which we call feasting in this chapter, and then when you don't eat enough, which is going to be our fasting. So starting off with feasting, when you overeat, we already know that when you consume anything in excess, it will end up being stored as fat. We don't have storage for protein, so when you eat more than your body has a reason to use, then it is going to be converted to fat for storage. Carbohydrates, we can store some of it as glycogen, but when glycogen is filled up, then the remainder is going to be converted to fat as well. So anytime you eat in excess, it's converted to fat. And now we're going to go ahead and take a look at how that is capable of happening. So when you take a look over here, this is a slide that we saw before, but just taking a look back at it, if you notice, if you take a look at acetyl-CoA, the step from acetyl-CoA to fatty acids is reversible. And so that tells us that if anything that can become acetyl-CoA can be made into fatty acids. If we look at the glucose going down the center, what we learned so far is it becomes glucose to pyruvate to acetyl-CoA and then goes to the TCA cycle. But if we have more glucose than we need and more than we can store, instead of going to the TCA cycle downwards, it's going to move to the right and be converted to fatty acids. Same thing with our protein. Our amino acids can become acetyl-CoA, and that means that they can be converted to fat when it is in excess. On the other end of the spectrum, we have fasting. Fasting is when we don't have enough nutrients. Now, remember we said that your body is going to be using glucose primarily for fuel. That's what the body prefers for fuel. And so when the glucose starts to be depleted in your bloodstream, it's because you're not eating, what your body will do is it will go to your glucose stores, which is glycogen, and it will start to use glycogen for fuel. But eventually that is going to start running low. So we're going to go ahead and take a look at what happens. Now when you start to run out of your glycogen stores, we're going to have to find another way to fuel the body. And this is again a review of what we've talked about before. If you remember, we said the majority of the body can run off of ketones, which are made out of fat, and then the brain and red blood cell specifically need glucose. So if you've run out of glucose, then your body's going to have to find a way to make it if you're not giving it to your body. And if you're fasting, we're assuming that you're not giving it to your body. So in that situation, your body's going to have to make its own glucose.
So if we did need to make our own glucose, let's look at what our options are. Now, if we can use fat to make glucose, that's always going to be our first bet. Remember, we mentioned in our previous chapters that we don't like to use glucose for fuel or to make glucose unless it's our body's last resort, because we don't have excess protein stored in our body. So we don't have a place where we can just go get some extra protein that's not needed and convert it to glucose or fuel. We are going to have to break down our own muscles or other tissues in our body that are made of protein and use that to turn it into fuel or glucose for our body. So that's not something that we want to do. So if we can avoid that, we will always avoid that. So we want to take a look at our fats first to see if that is an option. So taking a look at our fats, remember we said our fats are made out of triglycerides. That's most of the fat that come from our diet is in triglyceride form. And triglycerides are made with 1 glycerol and 3 fatty acids. We know glycerol enters right before pyruvate, so technically glycerol is capable of making glucose. So we could use that part of our fat to make glucose. But if we look at the fatty acids, they enter at acetyl-CoA, where it is not reversible. So we actually can't make glucose out of our fatty acids. Now again, think about it. The triglycerides are made of 1 glycerol and 3 fatty acids. So thinking of it that way, only a small part of the fat can be used to make glucose, because the rest of the fat is actually made of fatty acids, and those cannot be used to make glucose. So because of this, the body is not going to be able to keep up with its demands just using our fats, because only a small part of the fats can be used to make glucose, and that is the glycerol portion. So again, most of the fat structure, which is the fatty acids, cannot be used to make glucose. We can make a little bit of glucose using the glycerol unit that's in our fat structure, but that's not going to be sufficient enough to keep up with our body's demands. So at this point, we are desperate, so we are going to have to go over to our protein structures and use those to make glucose instead. So remember, we had some amino acids that were glucogenic and some that were not. So thinking about that, our glucogenic amino acids are going to be the ones that are going to be used in this situation, because that means that they are capable of making glucose. So for example, this set over here can enter at pyruvate. That means it is still reversible back up into glucose. So just to recap, for situations where we need to make our own glucose, we can technically do that with part of the fat, the glycerol part, but that's only a very small portion, and it's not going to be enough to keep up with the body's demands. So we're going to have to go to our last resort, which is breaking down our own body structures to get the protein that was in there, and using the glucogenic amino acids to make glucose for us. Now, that was how we would make glucose for the parts of the body that can only run off of glucose, but we said that most of the body can actually run off of something called ketones. If we take a look at how ketones are made, technically ketones are made from putting together 2 units of acetyl-CoA. I don't need you guys to memorize the full structures. I just want you to know that ketones are made from attaching 2 acetyl-CoA together. And we'll see why that's important in our coming slide. So now that we know that we use acetyl-CoA to make ketones, let's go ahead and look at our options here. Now technically all of our nutrients can make acetyl-CoA, which means they can all make ketones, but we're not going to be using the glucose to do that. We're not going to use our carbohydrates to make ketones, and the reason for this is if we had glucose, we would just use that for fuel. The reason we're making ketones is because we don't have glucose to use for fuel. So this over here would not be an option. That leaves us with our proteins that can make acetyl-CoA and make ketones for us that way, and our fats that can do the same thing. Remember, proteins are our last resort, because whenever we use proteins for fuel, that means we're breaking down our muscles. So if there's an alternate option, we're going to do that first. So right now, we do have an alternate option. We said we've canceled out glucose, but we still have fat as a possible option. So let's take a look at that. With our fats, our triglycerides, the type of fat that we find in our diet, is made of glycerol and fatty acids. The glycerol enters right between glucose and pyruvate, where it can technically go downwards and be used to make acetyl-CoA, and that way be used to make ketones. So we can use the glycerol to help us make more acetyl-CoA so that we can make ketones, but our fatty acid part of the triglycerides can also make acetyl-CoA for us. So all parts of the triglyceride structure, all parts of the fat can technically be used to make ketones. And if you guys remember, when we saw how the fatty acids make acetyl-CoA, we saw that acetyl-CoA is made out of 2 carbons attached to coenzyme A, and the fatty acid chain had a bunch of carbons attached to it. So we saw that we could easily break off 2 carbons each and make an acetyl-CoA, and then break off the next 2 carbons and so on, until we ended up with a bunch of acetyl-CoA just from that one fatty acid. So what that tells us is the fatty acids are actually able to make loads of acetyl-CoA for us. So they're going to be a great source of our ketone production because they can make tons of acetyl-CoA. So that's more than enough to be able to fuel the body. And so for that reason, we don't need to rely on our protein. Now, technically, these proteins are called ketogenic, meaning that they can be used to make ketones. And the reason for this, we now know, is because they make acetyl-CoA, and acetyl-CoA can be used to make ketones. So that explains why these were referred to as ketogenic. And technically, they can do that. But since we can sufficiently make enough ketones with our fats, we're not going to need to rely on these ketogenic amino acids to do that for us.
Now this here is a good slide to look at for review. It shows you what happens in each step, starting with feasting, and then short-term fasting, and then long-term fasting. So let's go ahead and take a look at what happens. At the very top, we have feasting, when you overeat. And we have carbohydrates, fat, and protein. In the carbohydrates bar at the top, it shows that some of the glucose is going to be used, some of it is going to be stored away in your glycogen stores, and then the rest is converted to fat. If you notice, the protein is the same thing. Some of the amino acids are used to make body protein structures, and then the rest of it is stored as fat. So that's what we already know about what happens when you overeat. Now, on the other end, we have fasting. So let's say that you have gone a few hours between your meals. That's what we call your short-term fasting. So about 2 to 3 hours between meals or so is short-term fasting, and we go through that on a daily basis. Now, with the short-term fasting, remember we said when you aren't actively eating, your body will use your stores for energy. So that's what you see happening here. In the blue bar, we now have our glycogen stores from the liver and muscles that are going to release glucose so that they can fuel our body. Now, the body isn't going to want to use up all of that glucose to fuel the whole body, because then we're going to run out quickly. So the body will also use some fat to fuel the other cells in the body that don't necessarily need glucose. Now, the one thing that you'll notice is that there is not a pink bar in the short-term fasting. The pink bar is supposed to be protein, and in short-term fasting we don't see it there. And the reason for that is because protein is something that we don't want to use for energy unless we're absolutely desperate. Because if we use it for energy, that means we're breaking down our muscle. So in between your meals, 2 to 3 hours, you still have enough glycogen that you can use for energy. We're not desperate yet, so we're not going to use protein. Now, when we go on to our long-term fasting, so about 24 hours or so without food, at this point, your body is going to have run out of glucose, which is why you don't see a blue bar there anymore, and it's going to have to find another way to fuel your body. Now, the thing that you'll see here, we have the body protein first. That's going to be any of your tissues, your muscles that the body can break down and use the amino acids from those structures to convert them into glucose. Now, some of those amino acids are not going to be glucogenic, so they won't be able to make glucose, but those we can go ahead and convert to ketones. And then our fat can be broken down into plenty of ketones, so those are going to be what are used to fuel other cells in the body that don't require glucose. Now we're going to go ahead and take a look at how alcohol is metabolized in the body. Since it's not a nutrient, we hadn't discussed it yet, but we do want to know what the body does with it. So starting off, we know that alcohol is something that can have potential health benefits. We did say it is considered a toxin, but it also can have some health benefits, in particular red wine. But those health benefits actually don't kick in until you're at least 35 years old. So any younger than that, and alcohol doesn't really have benefits at all. Another thing is that these benefits come with drinking in moderation, which is 1 drink per day for a woman and 2 drinks per day for a man. This doesn't depend on how quickly they're drinking or whether they're eating or not. It's always going to be 1 drink for women, 2 drink for men. You can't save them up and use them all on one day. Now, along with that, we want to know what is considered 1 drink. And it depends on the alcohol that you are consuming. If you're consuming beer, it's going to be 12 ounces. If it's wine, it's going to be 4 to 5 ounces. And if it is liquor, it is going to be 1.5 ounces. Now we had said that the nutrients need to be digested or broken down into small enough pieces before they can be absorbed. Now that is the case with the nutrients, but not with alcohol. Alcohol actually doesn't need to be digested in order for it to be absorbed. It can be digested if it hangs around in the body for long enough, but it doesn't need to be. Now, if it does hang around the body long enough to be digested, the first enzyme that's going to begin digesting it is called alcohol dehydrogenase. Now, this enzyme is actually found in higher quantities in men than in women. It's just the way that it works. And so because of that, men can handle more alcohol than women can, and that's why there is that rule of 1 drink per day for women and 2 drinks per day for men. Because the men are going to have more of the alcohol broken down, and so they're going to have less of the influence of alcohol. The other thing that I want to mention about that is if you are drinking on an empty stomach, the alcohol is going to go into your stomach and it's going to easily make contact with the stomach walls. Once it makes contact with the stomach walls, it's going to be immediately absorbed into the cells of the stomach wall. So it's not going to get a chance for alcohol dehydrogenase to break it down. But if you are drinking on a full stomach, the alcohol is going to go down into the stomach, mix with the food. The food is going to get in the way of the alcohol making direct contact with the stomach walls, or it will at least slow it down. And so that way, the alcohol dehydrogenase enzyme in the stomach has time to make contact with the alcohol and break it down. That's why when you drink on an empty stomach, you tend to feel the effects a lot quicker, because the alcohol is going straight into your bloodstream through the stomach wall instead of being broken down first. Now, if it does get through the stomach and get broken down, it's going to make its way into the small intestine, and that's where we have another enzyme that is going to continue its breakdown, and I'm going to go ahead and show you this through the next diagram. So here we have the different steps. We start off with alcohol, and we have the first enzyme in the stomach, alcohol dehydrogenase. And then we have the second enzyme from the small intestine called acetyl dehydrogenase. That's going to complete its digestion. Now it is going to then turn into acetyl-CoA, which has 2 path options. It can be turned into fat or enter into the TCA cycle. But alcohol actually prefers the route to fat, and so usually alcohol will just be converted into fat and stored that way. Now, if you are drinking too fast and your body can't keep up, you are actually going to start using a different system to process alcohol called the microsomal ethanol oxidizing system. It is part of your liver, and it is supposed to deal with any kind of medication, drugs. It is supposed to metabolize those. But if your body can't keep up with the amount of alcohol you're consuming, it's actually going to put the drugs on hold and start metabolizing the alcohol. Now, the problem with that is if the system is too busy dealing with alcohol and doesn't break down the medication instead, the medication is just going to enter the bloodstream as a whole, and you're going to have a much larger dose entering your body at once than the doctor intended for you. And that's why it usually says don't consume with alcohol on a lot of medications. As for other effects that alcohol can have, it starts off affecting the frontal lobe of your brain, which is what controls your judgment and your reasoning. After that, if you continue drinking, it'll affect the midbrain, which is what controls your speech and your vision. And then as you continue drinking, it's going to affect your cerebellum, which is what controls your voluntary muscle movements, your coordination. And then lastly, your respiration is going to be affected, which is regulated by the pons and the medulla. And that is something that can actually lead to stroke and death if it reaches that point. Another thing alcohol can do is cause malnutrition, and it can do this in several ways. First, we have certain vitamins, especially your B vitamins, that can't be absorbed when alcohol is in the way, so it can prevent certain nutrients from being absorbed. The other thing is that alcohol contains calories. If it's just straight up alcohol, we already know it's 7 calories per gram, but if it's a mixed drink, you have to think about what else is added into that drink and count those calories also. That can be something that can add up and lead to weight gain, obesity, and that comes with its own concerns. Now, the other thing that could happen is maybe you're taking into consideration the calories, and so you say, well, I'm not going to eat as much today because I want to leave room for the alcohol that I'm going to drink later on tonight. Doing that on a regular basis is going to displace the nutritious food that you're going to eat for empty calories from alcohol, and that can easily turn into a deficiency.